Hello everyone, welcome back to my channel Capsule. Let's begin by understanding what an endospore really is. In simple terms, an endospore is a highly resistant dormant structure formed by certain bacterial cells when faced with environmental stress. It is essentially the bacterial cell survival capsule built to withstand conditions that would otherwise be lethal to ordinary cells. Unlike reproductive spores in fungi, bacterial endospores are not involved in multiplying the population. Instead, one vegetative cell produces one endospore, and when the conditions improve, that spore germinates back into one vegetative cell. So it's a one-to-one transformation, not multiplication. Endospores are mostly formed by gram-positive genera like Bacillus and Clostridium, which include many medically important pathogens. So the significance or the importance of endospores lies in their incredible durability. They can survive boiling temperatures, desiccation, radiation, extreme pH, and even many disinfectants. In fact, viable spores have been recovered from ancient mummies and amber, surviving for thousands of years. This resistance makes them a huge concern in clinical settings as they are not killed by routine cleaning or antibiotics. Let's now contrast bacteria that can form spores with those that cannot. Sporulating bacteria like Bacillus subtilis or Clostridium botulinum form endospores when resources are scarce. These bacteria can lie dormant in soil and or dust for many years, waiting for the right moment to reawaken. On the other hand, non-sporulating bacteria like E. coli or Staphylococcus aureus lack this capacity. When conditions become unfavorable, they die off unless they adapt rapidly. Under the microscope, the position of the spore inside the cell offers important diagnostic clues. A central spore divides the cell equally and is typically of Bacillus anthracis. A subterminal spore which forms just off-center is seen in Bacillus subtilis. In contrast, terminal spores appear at one end as in Clostridium titeri. And sometimes the spore is so large it swells the cell at the one end called the terminal swollen spore, commonly seen in Clostridium botulinum. So these morphologies are not just academic, they help microbiologists identify pathogens in the laboratory. The endospores resistance mechanism lies in their multi-layered armor. At the core, it contains the bacterial chromosome, enzymes, ribosomes, and dipicolinic acid or dipicolinic acid, which forms calcium chelates. These chelates stabilize the DNA and remove water, reducing metabolic activity to almost zero. Around the core is the inner membrane and germ cell wall, which becomes the vegetative cells wall upon the germination. Next is the cortex, a thick peptidoglycan layer that ensures dehydration. Then we have the spore coat made of several layers of keratin-like proteins. This is the barrier to chemicals and enzymes. Finally, the exosporium is the outermost membrane, often involved in surface interactions and immune evasions. So, sporulation process is not just random. It's a regulated step-by-step process triggered by nutrient starvation. First, the bacterial DNA forms an axial filament. Then an asymmetric septum divides the cell into two parts, a larger mother cell and a smaller forespore. You can see in the image. The mother cell then engulfs the forespore, wrapping it in a double membrane. Inside the protected chamber, the cortex and the coat layers are built. As the spore matures, dipicolinic acid accumulates, dehydration increases, and resistant properties are acquired. Finally, the mother cell lysis, releasing the fully matured endospore into the environment, ready to survive indefinitely. Because spores are highly impermeable, we use a special staining technique called the Schaeffer-Fulton method. It uses heat to drive malachite green into the spore, followed by saffronin to counterstain the vegetative cells. This results in green endospores inside pink or red cells, making them easy to distinguish under a microscope. This stain is often used to confirm the presence of spore-forming pathogens in clinical laboratory settings. In medical microbiology, endospore-forming bacteria are among the most dangerous human pathogens. Think of Bacillus anthracis causing anthrax, or Clostridium tetani leading to deadly muscle spasm in tetanus. C. botulinum causes flaccid paralysis via one of the most potent toxins known to man. Even in hospitals, spores from C. difficile can contaminate surfaces and resist disinfectants, causing recurrent infections in the patients. This is why sterilization protocols in medical settings are designed to destroy spores, not just kill the bacteria. In fact, biological indicators used in autoclave validation are spores of Geobacillus stereothermophilus, because if you can kill this, you can kill anything. So now we have understood what endospores are, how they form, their intricate architecture, and their clinical significance. But here's the real twist. How do they survive such extreme conditions? What's their secret to being virtually indestructible? In this video, we will in the next video, we will dive into the biochemical and molecular mechanisms of resistance and explore how spores germinate back into the life when the environment becomes favorable. So hit the like button if you have learned something new. Subscribe if you are serious about mastering microbiology, and turn on notifications so you don't miss the second part of this exciting topic. Thank you for being with me.